Good evening, everyone. Hope you had a very nice day. Down my own area, my day went well, though the weather was very cool and I almost left off during afternoon class. Everything is fine. Okay, so today, quickly, we have lost 7 minutes already. We'll be looking at cell division and genetics. Okay, cell division and genetics. So we'll be also looking at under the cell division and genetics. We'll be looking at four subheadings: mitosis and meiosis. And I believe this is not the first time you are hearing about these two words. Then we'll also be looking at chromosome, genes, allele, then Mendel's law of inheritance, practical. Then we'll also be carrying out a few practical. So the practical aspect is first of all theoretical. Now it's left for you to do what? Conduct the practical yourself. Okay? So we are going on right away. So first of all, introduction to cell division. What is cell division? Like I've always encouraged you guys, whenever you see something, see a term or anything at all, you should ask yourself question, what is this? Then you go ahead and ask your question, why is it necessary that I must learn this? Okay. So what's cell division? Now, first of all, cell division. So it's made up of two words, right? Now, a cell was undergoing a division. What's the meaning of cell? Like we all know, cell is the basic unit of life. So what's division? Division means what? Being divided into two, right? I just use the same word to explain the word. Okay, so division means what? Replication. In some contents, you can say separation. Can I use the word separation? Okay, so I think we'll go with, okay, you can say being cleaved into two. That's cleavage. That's separating something into two. Okay, so when we talk about cell division, is what is the entire process by which a cell what reproduces, a cell what replicates. Okay, so cell division what involves what a single cell dividing to produce two identical cells. Sorry, cell division means what a single cell dividing to do what to produce two cells. Now, there are two major types of cell division, and that was the first thing we mentioned when this class started. First of all is what? Mitosis. And the second is what? Meiosis. Now, before we go ahead to look closely at, wait, hope I'm not too fast. Please, okay, if there's anything you want to tell me, please don't use the reaction button or the comment section because I'm not seeing them when I'm sharing my screen. You can just unmute your mic, take permission, and then ask your question, okay? Or if you're not hearing me, or if I'm not audible enough, you can also indicate, all right? Okay, so I'll continue. Let me try and be much more slow. Okay, so before we go into looking at mitosis, meiosis, how they occur, why they are even important to occur, let us look at cell division generally. We have agreed that there was process by which the cell reproduces, the cell what replicate, the cell what divides. So now first of all, cell is basic unit of life. And when we talked about, I think last 3 weeks ago, when we talked about laws of, when we talk about or something mentioned about what laws governing cells, we talked about that every that the cell makes up of what, sorry, let me get myself together. We talked about that what every living thing is made up of what cell. We went ahead to say that the cell is what basic unit of life. And then the third law there was that what every cell is what made up of pre-existing cell. So since no cell will just appear, now it means that for any other cell we see, it is made up of what existing cell already. So how then can you say that a single cell, this is a skin cell now, a single skin cell now what replicates to form another one? That's what we want to look at now. Okay, so why are cell division necessary? Why are they important? Number one, cell division is important for what? Growth. What is fertilization? Fertilization is what? Fusion of male and female gametes to give what? A single totipotent cell. Now, This single cell eventually gives rise to what? Someone like you, very fearfully and wonderfully made, and someone like me that cannot what take pictures and post on Facebook because I'm not as beautiful as you. Are you getting me? I'm not as handsome as you. Hope you guys are still with me, right? So, how then does this single cell called a zygote give rise to a whole individual like you? Cell division. So, through the process of cell division, what accomplish what growth. We what grow. All right. Now, one of the other importance of cell division is what in body repair. Imagine probably few years back, you are so you are so how like nowhere was entering you. Is that you are jumping from one table to another, or you are building houses in your parlor using wrapper and all. Now, peradventure, as you were playing, you mistakenly probably you fell down and then you scratched yourself and then you bled, and mom was like, why always careless or so? Now, if you notice something that whole day, first of all, apart from you being so quiet and calm because you have injured yourself, give yourself 3 4 days to 1 week, that injury will start healing up. Probably that small cut, the next thing you see, that cut, there's a fibrin clot covering it. Before you know it, that blood clots there, you peel out, and then you see a new skin. Sometimes it's whitish, before you know it is sometimes it's black, right? Now, how does that little cut, how is it that that little cut, next time you saw it again, it has already closed up? Remember, no cell can just probably appear. Every cell arises from pre-existing cell. The process is what? Through cell division. Now, cell division is also what? Important in repair. Maybe you cut yourself while trying to cut a green. Cell division what? See through that your skin cells what? Cover up. Hope you guys are still with me. And also, most importantly, we see cell division what? In the area of reproduction. Just like I mentioned earlier now, division of male and female gamete to give us to give rise to a zygote that undergoes cell growth to what? Cell growth, cell division, growth and differentiation to form someone like you, fearfully and wonderfully made. Now, there are also many other importance of cell division, but these three are the key ones. Okay, so now we'll be looking closely at these two types of cell division, the mitosis and meiosis. Hope you are still together, right? So
Yes.
mitosis, right? Thank you for the feedback. Mitosis, just like I say, this is not the first time you are hearing this word. So what is mitosis? Mitosis is a process by which a single cell divides to produce two other identical cells. First of all, cell division is what? A process by which your cells reproduce, right? Now we are saying there are two ways that these cells can reproduce and give rise to another one, to another cell. Is that these cells undergo a process called mitosis, or they undergo another process called meiosis. We want to look at the peculiarities of the two to see where they function, to see where they are important, to see where they are needed. So we are first of all looking at mitosis now. And I'm telling us that mitosis is what? A process by which a single cell replicates to give what? Rise to another cell. Now making it a total of what? Two. Now there is something peculiar about mitosis in the sense that whenever it occurs, it gives rise to what? Genetically identical cells. Now, if you look at the cell that gave rise to another one, if you are looking at the two of them, genetically, there is no difference. The exact copy of chromosome this one has is what replicated and transferred to the other cell. Okay. Now we are going to look at the stages of this mitosis, and also first of all, we also look at where it occurs in the human body and why it's necessary. Now, mitosis occurs in what is called body cells. Body cells. Now I can generally classify cells into somatic cells and what? Gametes. So gametes are what also called sex cells. There are cells responsible for what? Reproduction. Example, the spermatozoa, that's the sperm cell in male, and then the ovum female. These are what? Sex cells. Now, every other cell in the body is what? Somatic cells, or the body cells. So mitosis occurs in what? Every somatic cell. It occurs in every body cell, from your hair down to your what? Toenails. Are you with me? So peradventure, just like I used as an example, you were running and then you fell down and then you had what? A scratch, probably a tiny cut in your what? Kneecap, and then you bled, and then the skin there bled. Give it a week or plus to heal back. What happened? The skin cells there what? Undergo what? Mitosis. Now this enable it to what? Reproduce and what? Make up for those cells that were lost in the damage. Okay, so mitosis are what? Important for growth and tissue repair. That's their general function. Growth in the sense that the way you are when you are born, your body underwent what? So many cell division to give rise to what? You right now. Even the moment you are speaking, your cells are what? Still reproducing and replicating. It's also important in what? Tissue repair. That's the example we have been citing since this class started. Now, there is one thing I want you to note in mitosis, just like I mentioned earlier. Mitosis maintains the number of what? Chromosome. It maintains what? Chromosome number. In every somatic cell, we have what? Diploid chromosomal number. Okay? Now, that's diploid what? Chromosome number 2n. Now, after mitosis occur, this same chromosome number is what? Maintained, unlike in meiosis, just like we're going to see very soon now. Also, like I mentioned earlier too, mitosis what? Give rise to what? Two identical cells. So let's take a look at stages of mitosis. Now, the first stage in mitosis is what is called prophase. Okay, first of all, there are what? Four stages: the prophase, metaphase, anaphase, and what? Telophase. Now, I was actually expecting someone to have unmuted the mic and asked that there is one more stage that is remaining, but it seems like no one. Probably you guys are still looking at the slide or waiting for me to clarify. Okay, so in textbooks, in I think what's the name of the textbook used in secondary school? That textbook, I think they included one more phase called the interface. The interface. I remember the acronym I used during my junior hype maths: interface, prophase, metaphase, anaphase, and telophase. But that ends in secondary school. Okay. Now, in your university level, you are going to understand that the interface is not really a phase in cell division. Interface is normally called the what? Resting phase. Okay, don't worry, it's not here. Okay, if you are writing, you can write it down though. The interface is what called the what? Resting phase. Now, during the interface, the cell continues its normal growth activity, it continues its normal processes, and then it prepares for what? Cell division. So it is not what included technically in what? Division process. Okay. Now, let me give you what? Stages of the interface as well. Now, the interface has three phases: G1 phase, S phase, and what? G2 phase. Now, during the G1 phase, it's just what? Normal cell functions. During the S phase, what? There is synthesis of DNA. And then during the G2 phase, there is what? Cross-checking for what? Division process to start. Okay? Now, let's get back to what we are talking about: stages of mitosis. Now, there are four stages, just like I mentioned. I'll be looking at the prophase. Now, after the interface have occurred, the next thing we see is prophase. What happens during prophase? Now, during the prophase, the chromosomes won't condense. First of all, okay, now let me explain. In the nucleus houses the genetic material of a cell. Under normal condition, you cannot see the chromosome. What you see is what? A thread-like structure, a thread-like material called what? Chromatin. Now, during the prophase, the chromatin what? Condenses to what? Form the chromosome. During the prophase as well, the nuclear envelope what starts to break down, and then spindles are what formed. Okay, so now there are two key things that happens during prophase. First of all is what? The condensation of the chromatin to form what? The chromosome. Please, you can write this down. Condensation of the chromatin to form the what? Chromosome. Secondly, the formation of what? Spindles. Now, I'll tell you the what these spindles are used for. You see them in what? Metaphase, anaphase, and telophase though. Now, these spindles are formed from what is called what? The centromere. The centromere. Okay? Now, let's go down to the next phase, the metaphase. Now, during the metaphase, the chromosomes that have been formed, I think I should show you guys a picture. During metaphase, the chromosome, you notice that there is something like blue, then red, orange, green, and another blue lined up in the middle, right? Now, this is what? Metaphase stage. During metaphase, the chromosome comes and lines up at what is called the equator. Equator is what? A line that divides something into two. Now, the chromosome lined up at what? The equator and prepare for what? Migration. Are we together? Okay. So
Yes, sir.
let's go back to our slide. Now, during the anaphase, what happens during the anaphase? The sister chromatids are what? Now pulled apart and they undergo what? Separation. Now, after the anaphase, the last phase we now have is what? The telophase. What happens during telophase? The nuclear membrane is what? Reformed. And cytokinesis is what? Occurs. So let's go back to our slide. The image again. Let me explain from the image. Now, the first thing you are seeing there is what? The interface. Now, the interface showing for this chromosome. I want us to look at the interface very carefully. Can you identify the chromosome there? Are you seeing the chromosome there in the interface?
Yes, sir. Yes.
Yes,
Are you
sir. We
sure?
can see it.
Yes, sir.
In the
Yeah.
in the
It's
interface, you are seeing the
46.
chromosome.
No, we can't see it. Okay, no, it's not showing. It's not showing.
I was
Sorry, in the
I was
interface.
like I
It's
was
not
like
showing.
Jesus, this student have for my hand. Okay, very nice.
It's not
Okay,
showing. It's only written. It's only written.
exactly. Now, during the interphase, the chromosome is not visible. What you see there is what? The chromatin.
Okay.
I think I'm hearing myself. Someone's mic is on. So what you see there is what? Chromatin. The chromatin there is what? Diffused. Are you seeing those blue structures, right? The blue thread-like material. They
Yes,
can the
yes.
chromosome is not visible during the what? Interphase. Now, after the interphase, there is duplication of chromatin, there is duplication of what? Nuclear materials, and then condensation occurs. Now, the next phase is the prophase. The chromatin has now condensed to form what is called the what?
Sir,
Chromosome.
excuse me, sir.
That's what you're
Please, you
seeing
are
now.
too fast.
Hello?
You are too fast.
Okay, I'm
Slow
too
down a
fast.
bit. I'm right. Yeah,
Okay, so sorry. Thank you very much.
no
Now I have to slow
problem.
down now. All right.
From that interface, you said during the interface, the
Yes.
chromosome is diffused, which means what?
The chromosome is diffused. Diffuse means it is spread apart. That is spread apart so much. It is not, when you say something is condensed, that thing has what come together. When you say something is diffused, you can say the thing was spread apart. It is far apart from each other. Okay. So now during the prophase, each chromatin that is spread apart starts to condense. Okay. They start what? Condensing. They start coming together. Now, once they
Sorry, sir.
condense again, okay,
Sir, is it chromatid or chromosome?
chromatin.
Okay, sir.
All right. Okay, so during the prophase, the chromatin starts to what? Condense together. Once they condense, it now what? Becomes visible. Now, this thing we have seen now is what is called the what? Chromosome. Also, mitotic spindle start to what? Form. Now, if you look at the image labeled prophase, look at by the left, you will see something like thread line. Thread line was starting to form. It is labeled early mitotic spindle. Now, this mitotic spindle what starts to what? Form. It is forming from a structure called the what? An organelle called the what? Centrosome. Now, after it is formed, let's now look at the what? Metaphase. Now, at the metaphase, chromosome now what? Okay, first of all, look at there's a little small stage for the metaphase. So it's for your higher classes. It's called prometaphase. Now, during prometaphase, it's just transition between prophase to metaphase. Prometaphase is just like transition between prophase to metaphase. So during prometaphase, the chromosome is what? Trying to arrange itself in the middle line, in the equator. Now, at metaphase, they have what? Arranged themselves. If you look at the metaphase now, you see how the chromosomes are what? Arranged there very beautifully. Okay, now, during the anaphase, the chromosome now starts to what? Migrate. The
Sir,
spindle
please
what?
calm down. No.
All right, so let me give you guys 5 seconds to copy them. You can do this. 1 hour still remaining. Though
Please,
you can overshoot.
someone
Sorry?
should help me take screenshots so I'll send it to the group.
All right, so
Okay, we will.
okay, no problem. Thank you very much. During anaphase now, the chromosome that have been lined in the
Sir, please,
middle.
sorry. You said during the metaphase, the chromo what? Sorry, I'm hungry. Can I like repeat
Okay,
the line that you said about the
during the metaphase, the chromosome what lies in the what equator. Okay, let me open the slide so you can see it carefully. During the metaphase, the chromosome aligns what at the equator. You can screenshot this too. The chromosome what aligns itself at the equator.
I can't see this. I can't see that screen.
Now,
Okay.
a chromosome. Now, in chromosome, there is something in a chromosome called what? A Let me find an image of a chromosome and show you. Okay, now please just look at this chromosome now. Now, this chromosome is actually what joined by what? Now, during the process of cell division, the chromosome replicates. It duplicates to form sister chromatids. Chromatid. Are you seeing this bee-like shape? Now, these are what two chromosomes joined together. A single chromosome. It's formed what is called a sister what chromatid. Hope I'm making sense. This
yes, sir.
chromosome now have what, two parts, two sister chromatids, right? Now let's go back to our image, what we were discussing earlier. Now looking at this in anaphase, these two sides now, these two sister chromatids, they start what getting pulled out together. You are seeing how they are what moving to each opposite end, right? Now, as they are migrating towards the opposite end, the cell is now what preparing to what divide into two. You now have early anaphase, late anaphase, and then your what? Telophase. Okay? Now, during the telophase,
Please don't be angry. What did you say? Say during the anaphase, the sister chromosome chromatid starts to do what? Sorry.
the sister chromatid is being pulled apart to the opposite end by the what? Mitotic spindle. They start to what? Separate. Okay? Now, in the
By the what?
telophase,
Sorry.
mitotic spindle.
Mm. Okay, sir.
Are we together? All right.
Yes,
So
sir.
now, during the telophase, the sister chromatids have what reached the opposite poles. Now they have what reached the opposite pole. Now the cell membrane what forms again. The nuclear membrane what forms again, and now what something called cytokinesis occurs. That's the division of a cell into two. If you look at this telophase now, you see that what a line is what forming in between it. This line what eventually partition this cell into two. And eventually you now have what two daughter cells. All right, so let me continue. Now during the telophase, you now see what cytokinesis occurs, and the cell is now partitioned into what two. Look at it very well. Every single cell there is what divided down to form what the two cells. Let me recap what I just said now. The single cell we once had, now at the end of these two cells, we now see what chromosome materials. The cells now divide to what form two new cells. Now, these two new cells are what identical to each other in every what area. If you look at these two cells now, there is nothing different. Are we together? So this is
Yes,
what
sir.
end. This is the what end of what mitosis. Now, let's take a look at the second stage, which is the second type of cell division, which is called the meiosis. So the meiosis, the meiosis is what a type of cell division that occurs where in what sex cells. So the meiosis is what response now. Where was I again? I think I was mentioning something. Okay, so meiosis is what
Yeah.
the kind of cell division that occurs in what sex cells. It's kind of cell division that occurs in what sex cells. Meiosis is the kind of cell division that occurs where in sex cells. Yes. So now something peculiar about meiosis is that what it gives rise to four genetically different cells. Are we together? After meiosis have occurred, it gives rise to what four genetically different cells. Remember, in mitosis, it gives rise to what two cells that are what genetically identical. Hope you are picking out the differences. In mitosis, two cells. Meiosis, four cells. Mitosis, cells are genetically identical. Meiosis, cells are genetically different. Shall I repeat myself? Everybody got it.
Yes, sir.
All right.
You can continue.
Everybody got it. Also, in mitosis occurs where in somatic cells, while meiosis occurs where in gametes. Gametes are, like I said, that was gametes simply means sex cells. Example, the sperm and the eggs. Okay, so mitosis what reduces the chromosome number by half. In DNA, the chromosome number remains in mitosis, the chromosome number remains unchanged. That is, it is still what 2n. While in meiosis, it is what reduced to half. That's n. So now I was telling us the benefits of meiosis and we made mention of growth, cell repair, reproduction as well. Now, sorry, we made mention of what growth majorly and what cell repair. Now, in meiosis promotes what is called genetic diversity. Meiosis is responsible for the reason why you somehow resemble your mom and at the same time you resemble your dad. I'm making sense. Okay, I for one now, I have the height of my dad. I'm as tall as him, though I'm now taller than my dad. But now I have the what calmness and softness of my mom. Nobody should stress me though. By you getting me, my mom can withstand. There's this kind of person she is. She can withstand stress, anything at all. So I was took after her in that area. Now this is what meiosis in action. Meiosis what allowed me to what have that diversity. Is if it were mitosis that we were giving birth to, I would have resembled my dad totally in everything. But due to meiosis, I was able to have some diversity. I did not was take after my dad fully. Let that take after my mom fully. Rather, there was what? A blend. Now, I want us to what? Look at how this meiosis occurred. Stages of what? Meiosis. The stages is bound to be what? Different. Why? Because unlike mitosis, meiosis what leads to reductional division. So stages of what? Meiosis. Now meiosis occurs in two stages. First one is what called meiosis 1 or reductional division. Then the second one is what called what? Meiosis 2. Okay. So in meiosis 1, we have just the same 4 phases, so but now you're adding one
Excuse
to
me,
each and
sir.
every one of them.
Meiosis one
Okay.
equals is called what?
It's
Excuse
also
me,
called
sir.
reductional division. Now,
And
both
meiosis two is called what, sir?
duplication phase.
Thank you, sir.
Now,
Sorry,
Sir,
what
you
did you
said
say?
what?
Sorry, you said
sorry,
You said what? What you just
duplication,
said now.
duplication, duplication,
Okay.
doubling,
sorry. What is the first one called?
reductional. Both mitosis and meiosis are the same phases, the same 4 phases. Interphase, prophase, same 4 phases. And remember, I told us that interphase is not really a part of division. Interphase is like what? The preparatory phase. Just in case you should still see interphase. Just know that interphase is just the preparatory phase. In standard division is not what considered stages of what cell division. Okay. Now, for the prophase, metaphase, anaphase, and telophase, you now be adding one to them. When we enter second stage, we're now adding them two. That's prophase 2, prophase 1. Every other thing remains the same except key things that changed. So in prophase 1, there is also what condensation of what chromatin to form chromosome. Now, after this happens, crossing over. Are you getting me? Crossing over what occurs in prophase 1. God will have to help us now. If I were to be in a class now, I would have been drawing in the board. But since I have no board here with me and it is night as well, I think I'll have to do justice by explaining this. Now, in prophase 1, something happened. The chromosome, the chromosome formed what? Okay. Where will I come from now? Okay, let's start this way. All right. After the chromatin is condensed, it forms what? Chromosome. Now, each chromosome locates its homologous pair and binds together to form a tetrad. Okay, hope you guys are still with me. Now,
Yes, sir.
each homologous chromosome binds together to form what? A tetrad. Now, crossing over occurs. What is crossing over? Crossing over is what? Okay, let me use an illustration. Will it be possible? I think I have an image too. Okay, can you see this image?
Excuse me, sir. Please, the binding of the homologous cell. Did you say it formed tetra?
It is called tetrad. T-E-T-R.
Okay, tetra.
Tetrad.
Sorry, spell it
Tetrad.
again.
T-E-T-R-A-D.
Yes, sir. Okay. Thank you very much.
All right. So now each okay, please get me now. Okay. Now non-sister chromosome, non-sister chromatid. Sorry, non-sister chromatid does what? There is some sort of shuffling. It crosses to the what? Next homologous chromosome. Are you still with me? Okay, take a look at this image now. Now, do you notice something from these two now? from these two chromosomes. Crossing over have occurred. What is this crossing over? A chromatid, a sister chromatid from this chromosome moved to the next chromosome, and the sister chromatid from this chromosome came to this one. This what creates what is called genetic diversity. Is it clear?
No, sir.
Okay, thanks for your honest answer. Another person lost. Alright, let me explain again. Crossing over is what? A situation whereby what? Non-sister chromatids does what? Exchange their genetic materials with the other what? Chromosome. Now, I want you to use this image to what? Picture this now. This chromosome what? Pairs with what? The other homologous chromosome. Now, when this happens, the sister chromatids of now this guy is called, see this guy I'm going to mark now. This one and this one are called what? Sister chromatids. Now, these two, okay, let me come in again. From this my image I have here, this image I have here now. If you notice that there is this section now moves to this part. There was what exchange of genetic material at these two ends. Now, this exchange of genetic material between these two chromosomes is what is called what? Crossing over. Have you gotten it? I should repeat
Yes.
myself. Okay. Let me come again, just for the sake of clarity. Now, these two chromosomes are called homologous chromosomes. They locate each other. Homologous chromosomes are what? They locate each other, and now crossing over occurs. They now what? Exchange genetic materials between what? Sister chromatids, between non-sister chromatids. Alright. So let's get back to our first stage again. Now, after this have happened, the second stage occurs, metaphase 1. This homologous chromosome what? Aligns at the equator again. They aligns at what? The middle. You know what happened in anaphase? The homologous chromosome are what? Separated from each other. Now, these what? Finally, we now have about telophase 1. Two haploid cells are formed. Let me go back to the diagram. Diagrams did not really do much justice to it. Now, this homologous chromosome arranges at the equator. And now what? This the diagram is actually poor. It did not help much. Okay, let me try and explain. Now, these two chromosomes called the homologous chromosome, they are arranged at the equator. Okay, so they're now what? Separates. After the separation, we now have two haploid cells at the end. Are you guys still with me? Any question?
No, sir.
No question. Okay, wow, that's very nice. Okay, so you guys are following. Now, the second phase. Now, this second phase is similar to mitosis, just that no chromosome duplication occurs. Now, what happens is that what? Just every single phase that happens in mitosis is what happens here. The same prophase occurs. The two cells comes together again, condenses their genetic material, metaphase occurs. The what? Each homologous chromosome now what? Gets lined up in the middle, and then what? Gets pulled apart. Now, at the end of separation, instead of now having what? A cell that's having two cells that are genetically identical, you will now have what? Four haploid daughter cells. Now, look at this diagram now. Look at image labeled number 1. Now, this is the interphase. After this, there was what? First division to give us what? Number 3. Look at this image labeled number 3. This is what? The end product of first meiotic division. After this, the cell now what underwent another division to now give rise to what? 4 haploid cells. If you look at these cells carefully, you notice that they are not paired again. Each of them are what? Single. That's what? So sorry. Each of them are single now, and it's called what? Haploid cells. Let me take us back to mitosis and show us something there. At the end of mitosis, okay, the cells is not condensed. If you look at very well, you still notice that the chromosomes here are paired. But at the end of meiosis, the chromosomes are no longer what? Paired. They are now what's called haploid chromosomes. That's half. They are now called what? Haploid chromosomes. I summarize the whole changes here. Okay, someone says she wanted to snap. Please, you can snap it now. Meiosis 2. Okay, now I summarize the whole comparison here. Comparison between mitosis and meiosis. Now, mitosis have what? 1 division. While meiosis have what? 2 divisions. In mitosis, we have 2 identical cells. In meiosis, we have what? 4 different cells. Mitosis plays a role in what? Growth, repair, development. While meiosis plays a role in what? Reproduction. Just reproduction. Nothing more. It only performs what? Reproduction. Now, also in mitosis, the number of chromosomes are what? The same. While in meiosis, the number of chromosomes are what? Halved. I think I mentioned all this before, right? Okay. So we are going to move on to the next topic we have now. I hope we are going to do justice to it in 30 minutes. Chromosome. Now, I believe, was it last week? No, it was not last week. I think last week or so. Talked about DNA, we talked about macromolecules of life, right? Now, I mentioned to us that the way DNA is packed in the nucleus is that each DNA is what? Intertwined around what? A protein called a histone protein. So, the packing unit of what? The DNA is now what is called a chromosome. Now, if you look at this image I have here, you see how long the DNA is. But this actually extends very long.
Excuse me,
It
sir.
can
You said the packing what is called chromosome.
the packing
I said
unit of the
chromosome is called
DNA.
what?
The packing unit of the DNA is a chromosome. Okay. So the chromosome comes into play because there is every need for this. If you look at this DNA, you see how elongated it is. There is every need for this thing to us packed in such a way that it can fit properly into the nucleus without disorganizing. So each DNA is what? Wrapped around a protein called a histone protein. I can remember spelling the histone protein during that class. Histone. Now, after its packing, you now have a structure called the what? Chromosome. Okay. Now, in humans, we have what? 46 chromosomes of 23 pairs. Now, each pair is inherited from what? Sorry. Now, we have 46 chromosomes, 23 pair. Now, each 23 is inherited from the what? Either parents. You have 23 from your dad, 23 from your mom. When these two cells fuse together, it gives rise to what? You, a zygote, which what gives rise to everything you. Now, also, the chromosomes are found in the nucleus of eukaryotic cells. Okay, chromosomes are found in what? Nucleus of eukaryotic cells. Remember, in prokaryotic cells, what we have is the DNA, which is found in a region called the nucleoid. Prokaryotic cells does not have nucleus. Okay, so lastly, okay, let me close this thing. So lastly, the chromosome what? They bear the genes which determines the traits. They bears what? Gene which determines traits. What is traits? Traits are what? Inheritable characteristics. Just like I told you a lot of us that I'm very, very tall, right? Exactly. So I inherited this tallness from what? From who? From my dad. I'm not saying my mom is short. All right. So genes and allele. So what is a gene? What is a gene? What is a gene? A gene is a unit of inheritance located on the chromosome. Now you can say a gene is what okay, I mentioned the unit of inheritance. You can say a gene is what? A sequence of code that what defines a threat.
He said we can
Can
also
continue.
say that a gene is what?
All right. So I think you can also say that the gene is what? A sequence of instruction that defines a particular threat. Now, I wanted to give us an analogy. Imagine you have, I don't know how many of us here are chefs that can cook, that can probably, I won't say cook more than I do now, but can at least try to cook just like I do. Okay, now, and I want to believe that you have a cookbook. Probably you have seen a cookbook before, a book that they have ingredients and everything written inside it. Now, a cookbook can contain, let's say, 10 to hundreds of dishes. That's how to prepare so many food. Now, one thing about that cookbook is that what is written, right? Is written in words, right? DNA and all. Now, your cookbook is what is written in sentences containing what different dishes and ingredients, procedures, and how to do, right? Now, let's say that every chapter is a dish. Now, the book now has what 100 chapters. Now, if you say that if I want to cook a egusi soup now egusi soup now in that cookbook can be chapter 23. Now, everything in that chapter 23 is what information on how to prepare egusi. Now, if I should carry that chapter 23, read it and effect it very, very well, I can replicate the egusi I'm going to see in the picture there. I can replicate it inside my own pot. Are you getting me? This same way is how the genes and chromosomes work. Now, the chromosome is just like your cookbook. The chromosome contains what? DNA materials, which contains what? Information, sentences, grammar, add this, add that, remove this, remove that. No, add salt, no, add one sachet of add 1 cube of Maggi. Are you getting me? So your chromosome is like your cookbook, which contains the instruction to what? Give rise to a human like you. Now, a particular segment of that chromosome, when you read it out, now in that your
Yes.
cookbook, you can see a sentence. Thank you very much for the feedback. You can see a sentence. A sentence will now say, add this to this. Are you getting me? Now, that whole chapter in that cookbook, that whole chapter in that your chromosome, that should tell your body when you are developing, I want this girl to be very fair. That is called a gene. It is now what a gene for being fair. Are you getting me? So it's a sequence of instruction that defines a particular threat. I think this definition is very straight to the point. So you can say a gene is what a sequence of instruction that defines the what threat. Threat, not not my grammar. I don't mean threat like I'll threaten you. Okay, I mean TRAIT. Not my pronunciation. All right. So allele. What's the meaning of allele? Allele means what? Variants of a gene. That's forms of a gene, the different forms of a gene. Now, let me explain as well. Imagine in that your cookbook, you have egusi with fish. That's for chapter 23. And then in chapter 24, you are seeing egusi with chicken soup. Hope you are still together. Chapter 23 was saying egusi with what? Fish. 24 was saying egusi with what? Chicken. Now, the two of them are the same egusi soup, but one was being cooked with what? Fish. The other one was cooked with what? Chicken. Likewise, in your chromosome, there is something called what? Variant of a gene. That's termed an allele. Now, a gene in that your chromosome, which is your cookbook, a gene can read this trait, this person should be what? Fair. Another segment of that chromosome can read what? This person should be what? Dark. Two of them are talking about complexion. Some of them are talking about egusi soup. But one was saying this person should be fair. That is this egusi should be what? Cooked with fish. The other was saying this person should be what? Dark. That is this egusi should be what? With chicken. Please, I don't know why I'm mentioning food. Somebody should not go and eat in the dream, okay? Okay, I'll stop using food. All right. Okay. So I hope you guys have gotten the point, all right? Don't mind the soup and all. And the point there is what? An allele is what? A
Yes,
variant.
sir.
That's a different form of what? A gene. Thank you so much. Now, the next point I want to mention here as well is that some allele can be what? Dominant. Some allele can be what? Recessive. I want to tell us now to do a practical with what our family, but I can't supervise this practical, so it will have to be on you. Now, okay, let me use my own family for example now. I am very tall in my family. But I used to tell my elder sister I should borrow you some of my height. Are you getting me? So I'm not saying she's short too. I'm not saying she's short too, but I'm saying I should was borrow her some of my own height. Now you can put in the words you want, okay? Now, for me, my God, how will I explain this to you guys to understand? All right, let me use another example. When you say a gene is dominant, not a gene actually, an allele is dominant. Wow. This is not going to be hard for us. Okay, let's start from allele first of all. Maybe we explain that. An allele is what? A variant of a gene, right? Okay. So we are going to use the gene to be tall, for example. Opposite of being tall is short, right? So if you are not tall, you are short. Now, an allele for being tall is capital T. If you have a bill, please write this down, okay? So you can easily reference something. An allele for being tall is what? I think I should also write here. Now, we are talking about being what? Tall. An allele for being tall is what? Capital T. An allele for being short is what? Small t. Hope you are together now. Beautiful. Now when we say that an allele is dominant, it means that if I should inherit this is from from my dad, this is from my mom. Okay. So if peradventure I should inherit this, let me start from the very beginning. Now, if now this is called the what? A genotype, a complete set of gene. Now, um, if I should inherit this capital T from my dad and this small t from my mom, and then I have something like T, this is my genotype. But then my phenotype, please, phenotype is what? Expression of what? The gene. Okay? Phenotype is what? The physical expression of what? The genetic material. What you are seeing. Now, my gene now is what? My genotype is capital T and small t. Capital T meaning tall, small t being what? Short. But then, phenotypically, if you are looking at me, I am very tall. Does it mean I don't have the gene to be short? No. Hope we are together. Is anybody lost?
No, sir.
Okay, everybody's
No,
following
no,
me,
no,
right?
we are together.
Thank you so much. Please, I want us to get this point, okay? Now, I just showed us a genotype of my dad, TT. That is what homos. Okay, let me explain some terms as well. When I say homozygous, I mean we're having what? The same allele. That's typical allele. If it's small t, two small t, homozygous recessive. Two capital T, homozygous dominant. Okay? Now, my dad is homozygous for being tall. My mom is homozygous for
Sorry, I
being
have a
short.
question.
Sorry?
I have a question.
Now you said
Okay,
homozygous is what?
Homozygous mean having identical allele. I think someone there wanted to ask a question too. So now I inherited from my dad this gene for being tall, and I inherited from my mom this gene for being short. So since I don't have identical gene, that is, I'm not identical for tall, I'm not identical for tall, I am now called what? Genotypically what? Heterozygous. Heterozygous, that means different allele in the same locus. Heterozygous means different allele. Now, this is my genotype. I have both tall and shortness inside of me. But now, if you're looking at me from outside, I'm a very tall young man. And they are probably asking, what happened? Is it that he's not inheriting the gene for being short again? So that's what we are still going to discuss next. Mendel's, I think second law was saying what some allele can be what dominant over the others. Now, looking at this genotype, you can see that what being tall is dominant. That is, if tallness and shortness are expressed in the same genotype, the tallness what marks the shortness. The person will now what grow tall. I don't know if you are getting me. Then you come again. Just like I've expressed this gene for being both tall and being both short. Since tallness is dominant in my blood, if you look in my gene, if you look at me from outside, I will now what still grow tall, despite having that what blood for what being short. Now, when they say a gene is an allele is recessive, they simply means what that allele cannot be expressed in the heterozygous format unless it's what homozygous recessive. Now I said an allele. Now this is my genotype, right? Um, I have both tall and short, right? Now, but if you are looking at me from outside, you see me as being tall. So I was saying that being tall is what dominant. So when the gene for being tall and gene for being short, two of them are what expressed together, being tall is going to max that of being short, and that individual is going to appear tall. Then I was saying when an allele is recessive, that allele can only be expressed in the homozygous format. Homozygous mean that allele that is recessive must be two for the person now to what exhibit that what trait. I say, let me show us an example. This is still me that has both tall and short in my blood. And this is now someone like my mom that is short. Person has both short and short. So if I should give birth to someone with capital T and small t, capital T, small t, that person is still going to be what? Tall. Why? Tallness is dominant. But if I should now give birth to someone that has small t, small t, small t, small t. Now, this is now what? Homozygous recessive. Now, shortness can now be what? Expressed. So, the person now looking at the person will now be what? Short. Despite the father is tall, oh, and the brother that was giving birth to is was tall, oh, but now this person will now be what? Short. Is it clear? Any question?
It's clear, sir.
Hope it's clear for everybody else. So I should now move on. All right. So Gregor Mendel. Now, Gregor Mendel, he was actually, let me tell us a story. I think first time I heard about this was in my primary six. A lot happened in my primary six though. Now, that was the first time I heard about Gregor Mendel, and I wanted to be a genetics. I was like, wow, this man did so many things. I love genetics. But, well, I'm not paying my school fees. My parents were like, it's medicine or nothing. And here I am. Okay, so Gregor Mendel was one of the first people to carry out experiments to talk about inheritance. trait. How is it that this young boy is as tall as his father? Or how is it that this your wonderful pointed nose is tempting your grandmother hard, though your mom does not have it again? It now reaches your own turn. You now come what? Somehow, you now have the pointed nose of your grandmother. How? How did it happen? Is it that the trait disappeared and then reappeared again? I heard a story in the olden days. They will say that was somebody reincarnation. They'll say that the grandmother probably came back to life again. Gregor Mendel, just like I was very skeptical about all this. So he set out a way to what find out what and what's really happened. Now he studied inheritance using pea plants. And we use pea plants because first of all they are easy to grow. Secondly, they can be self-pollinated, cross-pollinated, and also he can also what pollinate them by himself. And they also what have very contrasting characteristics. Some are tall, some are short. Some have white flowers, some have pink flowers. All right, so through this study of inheritance using these pea plants, he now formulated his own laws of inheritance. How? He observed patterns. He noticed that this is the tall plant, this is the short plant. When I planted the two of them, this pea normally always produce peas that are tall. This one always produce peas that are short. Now, he comes and pollinates this pea. Self cross pollination, right? Now, man can be agent of pollination. Now, you come and take pollen grains from this pea plant, go to the other pea plant, the short one, pollinate it. Then he watched so that when they now probably produced seeds, he took the seed, planted the seed, give it some months. Once it germinated, some seeds were tall, some were short. Then he was what? Surprised. He recorded down his findings. Now, those short plants again, he planted many of the short ones, the one that are purely short, took their seed, replanted the seed. All of those plants were short. He came to the one that were tall, took their seed, planted their seed. All of them were tall. Then he cross-pollinated them, planted their fruits. Some were tall, some were short. He recorded them down again. Now, those that were short, those that were tall, he cross-pollinated them. That's he used the two of them, the F1 generation, replanted them again. He was noticing different characteristics. Then he was spelling them down. So after everything, he now concluded that what traits are passed down as what discrete units. That is, traits does not just is not something complex. It's just that they are passed down unit by unit. Now, this discrete unit was what he called it. It was eventually that it was now named what gene. Remember, I told us that what genes are a unique set of instructions that defines a trait. So Mendel from his experiment was saying what this trait I'm seeing is just like a single sentence. It's just like an instruction for what? It's just like an instruction that was defines it. So this was what he founded out. This was what he was found out, and eventually it was established that what these discrete units Mendel talked about your what genes. So alright, so from his experiment, he concluded what he derived out what laws. I named them what Mendel's law. Are you seeing how selfish man is? So now, how many years ago? Yet his name is still what being held through his law. Now, Mendel's first law. Now, his first law is what is called law of segregation. That is, each organism carries two alleles per what trait. For being tall, even though phenotypically I am very tall, probably in my gene, I have the gene for being what tall and that for being what short. So now, during gamete formation, that's during the process of meiosis, the allele are what separated independently. The alleles are what separated. Okay. So now, each offspring will now receive what? One alley from each parent. Are you getting me? Let me come again. It's my drawing board. Now, this is me, right? Okay, I think I use red initially. Now, this is me. So, they were saying during gamete formation, no, meiosis will reduce this into what? Half. So, I'll have something like T here in meiosis and T here. So, each sperm cell will now have one capital T, one small t. Alright, so this is how sperm cells look like. So remember, I inherited this from my dad, and then this one from my from my mom. Alright, so Mendel was saying what? That alleles segregate during gamete formation. So this is what? A single, remember, this was what? A single chromosome. During formation of gametes, let's say sperm cells, it is now what? Separated into two. One for what? Being tall, one for what? Being short. And let's move on to the next law. Men, wait, was the first one clear? Did everyone understand? No
Yes,
question.
yes, we understand.
Alright, so
Yes,
let's move
sir.
on. Alright, thank you. Alright, so Mendel's second law. This is also called what? Law of independent assortment. Now, he said that what? Genes for different traits assort independently. Okay, let me explain. Assortment is what? Separation. It's also called what? It's called law of independent assortment means what? The way genes are shuffled, it is what? Independent of each other. Let me do it. Let me repeat my drawing again. Now, the best way I can explain this, I have many friends that understood the laws of the Mendel's law, but once it gets to this part, they get lost. Now, Mendel is trying to say that when traits are being what? Separated, when traits are being inherited, they are inherited independently of what? Each other. Now, imagine you want to travel. Let's say you want to go on a vacation. That's every student's dream, to go on a very long vacation. Now, imagine you want to go on a vacation now, and then probably a trip, a day trip. That's just a day trip to, let's say, Victoria Island. Alright, so this day trip you have to wear your best of clothes. Now, you have a box where you put shirts for guys, another box where you put what? Trousers. Now, for females, let's say you have a box where you put shirt. Now another box where you put what? Skirt. Now, the law of independent assortment is telling us that what? The shirt that you pick to wear does not influence the color of skirt that you choose to wear. Let me repeat again. First of all, you have a box or you have a drawer where you pack your shirt. You have another drawer where you pack your what? Skirt. Let's say in the drawer for shirt, you have a blue shirt and a red shirt. In the drawer for skirt, you have what? A pink skirt and a green skirt. Law of independent assortment is telling us that when you want to pick the clothes you wear, the color of the skirt does not determine the color of the skirt. Okay, that's the color of your top does not determine the what? Color of your skirt. So if I should open the drawer for what? Your top now, and I put my hand randomly, that's independent assortment, I pull out one shirt, and I notice that the top I pulled out is what? White in color, and then I wear white. I open the next drawer, I dip my hand, and I pulled out. What's the worst color blocking I would do now? Let's say I pulled out ash. I'm not going to say ash and white are not matching. That is black and white that should go perfectly. No. Once I pulled out that ash, I will wear it like that. That's independent assessment, that the inheritance of one allele does not influence the other one. Let's bring it down to normal genetics we are doing now, normal phenotype, since you can easily look at. Now, let's say the gene for being tall and the gene for height and gene for complexion. Let's say probably when the genes are being assorted to give rise to someone like me. Now let's say that there's a gene for being tall, is just tall and short. For complexion, it's just being fair and dark. The fact that I am tall does not mean because I am tall, I must be fair. So it's this same law is saying it's not because I am tall, that was why I am dark. No. This law is saying the two genes are inherited differently. They are inherited differently. It is not what affected by the other. I can choose to be tall and at the same time fair. I can still be tall and the same at the same time dark. This same law of independent assortment is saying, peradventure I am short. Because I am short does not mean you should treat everybody that is short. Let them be short and be dark. So they will acting devil in the church. Are you getting me? Now, you can see someone that is short and the person will still be very fair. So that's what this law of independent assortment is telling us. Oh God, God is very wonderful. I wonder if there is no such law as this. If they should say every person that is short must be dark. Hey, there will be problem. All right. Now, all these things increase this genetic varieties, genetic variation in offspring. Wow. All right. Okay, so we'll be looking at, first of all, that thing I was drawing that time, I've forgotten the name it's called. It's just used to determine genotype. But there's an easier way. Now let me redraw that stuff again. I was giving you guys my genotype, capital T and small t, and then that of my mom there, right? Capital T and capital T. Small t and small t, right? Then I wanted to cross for offspring. I'll show you something like this, right? Something like this, something like this, something like
Sorry,
this.
sir. Can I ask you a question?
Yes, go ahead.
What if you are an average person? What is your genotype?
Okay. Now, first of all, in real life, most times, now, first of all, they say that some genes are dominant, some are recessive. Some other time, the two genes can influence each other. That's, you can now have intermediate variations. This is not for a level though, but let me explain somehow. Being tall and being short, there is no far extremes. Okay, there is nothing like, okay, first of all, wow, where will I start from so you pick this up? I saw a puzzle. They asked a question: at which point will you continue pouring sand for that sand to become a heap of sand, or at which point will you continue removing sand for that sand to not become a heap of sand again? So likewise height, okay? Now, being tall and being short is what we looked at, and we now classify them. This one is short, this one is tall. But on a normal level, there is no extremes in the gene. I don't even know if I get what I'm trying to say now. Now, if I'm to ask your question, I'll use a simpler version. Let's use complexion for example. Why is it that some people are not at the extreme of being fair, some are not at the extreme of being dark? There is still interactions between the genes. Sometimes the genes are not totally dominant. Are you getting me? Now the gene for being tall sometimes is not totally dominant over the one of being short. So at some point there was arrived at the middle. There was an experiment men they conducted. I should have added this here too. Now there was an experiment men they conducted. Let's use white and which color are we going to go with now? White and yellow peas now. Now when okay. Which color of white and yellow give you now? Three primary colors, red, green, and blue. Okay, let's use red and blue. See, red and blue will give you green together, right? I think yes, that's true. So okay, yes, red and blue should give you purple, not blue, not blue. That's purple. Let's say you brought a red plant and then a blue plant. Let's say that blue is dominant, red is recessive. So let me be drawing. Now, capital R is what's dominant. That's red. Red is dominant over blue. Okay. Red is dominant over blue. Now, blue is what? Recessive. Now, when we cross these two, what are you going to get? You're going to get capital. I see this my diagram now. Okay. Now, when we cross these two, we are going to get capital R and small r. So, since red is dominant, this flower should be red in color. But sometimes there is something called incomplete dominance. Are you hearing me? Person that asked the question, there's something called what? Incomplete dominance. In the sense that there's what? No two extremes. So instead of being just red or just being what? Blue, they now come to the middle and stand. None of them is what completely pulling the extremes. None of them is what completely dominance over each other. Red is not totally recessive. Um, so red is not totally dominant. While blue is not what recessive. So they arrive at the middle. Red plus blue is what purple. I don't have purple color here. I would have colored this one purple. Am I making sense to you guys? Are you getting me?
Yes.
Okay. Now, likewise, let's use the same thing for height now. I think this is a simple example before you come to height. Let's say now there is a gene that should say every child born by this parent should not be above, let's say, 3 feet. And then another one was saying genes for tall should not be above what? Should not be below what? 6 feet. But due to incomplete dominance, they can arrive there at the middle. Can now be having what? 4 points, 5 points. Okay. Now, but then beyond gene, there are so many things as well that can affect height. Okay. So yes, gene can say this should be the cap of what someone should grow to, but it does not totally on its own define the height of someone. Just like genes are not totally responsible for what? Intelligence. So let's not go deep into those words. Let's remain surface level for now. Before that, I think I was trying to draw something. Okay, I was trying to explain the Punnett square, why it was important. Alright, so from this now, I'm trying to draw what? F1 generation of these two crosses. You notice, before I finish drawing this, it is more what? Tedious and stressful. So, Punnett designed a square that can help me with this task of drawing now. Something as cheap as this. So it is used to predict what both the genotype and what phenotype ratios. And it can be used for what one trait, that's two alleles. Now if you look at this, capital G means green, small g means yellow. Okay. Let's also say capital G. This is a flower that's showing here. Let's say capital G now means, I think we are going to use our own. Let's use our own. Capital G means growth, that's height, means tall. Small g means short. Capital G means what? Dark. Small g means what? Fair. Okay. Hope you guys are with me. You can write it down somewhere. Capital G means what? Let me help you guys here. Capital G means what? Tall. Small g means what? Short. Okay. So Punnett square can be used to easily cross two individuals. Let's say a parent is what? Both tall and what? Has the G for what? Tall and short. That's capital G, small g. Since tall is what? Dominant. This person is what? Phenotypically what? Tall. Now, another parent, this one here now, have what? Two G's for being short. So phenotypically, this person is short. So for me, instead of me to start drawing lines and all, I can easily cross them using this word Punnett square. I can say G, G, capital G, small g, G here, another G here. So that's one. That's capital G, small g, capital G, small g. Then another one, this G and then this one, you come down here and put it. This one and this one, come down here and put it. Are you seeing it? After this now, okay, I want to ask us a question. I want us to answer me, okay? Now, my question goes like this. Looking at this Punnett square now, how many individuals are tall? How many of the offsprings are tall? I'm waiting for our answer.
Two at all.
Two are tall.
Yeah.
Okay? Okay. Any controversial answer? No?
Just
Alright,
one, I think. Is it two?
just one. Okay? Okay. Any other
Two
answer?
at all, I think. Two.
Two are tall. Alright, so the correct answer is you guys are actually correct. The two of them are what? Tall. Why? The gene for being tall is what? Dominant. So if you look at it very carefully, we have four offsprings. Two are heterozygous, capital G, small g. Since G is what? Dominant, we have tall. Okay? Then, so how many individuals are short? How
Two
many individuals
individuals
are
are
short?
short. Two are
Yes,
short, sir.
two are short.
Two.
Alright, that's nice answer. That's very beautiful. Okay, so Punnett square can be used for what? Monohybrid cross. Monohybrid cross is where you are crossing two individuals. You are now looking just at one trait. Okay? Just like we are looking at the trait of being what? Tall or short. That is height. In this cross, we are just looking at what? Height. Also, you can use it for what? Dihybrid cross. Dihybrid cross. Dihybrid is what? Involves what? Looking at two traits at the same time. Let's say you are looking at what? Being tall and being fair at the same time. So it's a very complex, it's a very complex what? I think you are going to use this 1039. Time. So it is used to demonstrate independent assortment. That thing I was trying to explain to us, that being tall does not mean you must be fair because you are tall. Neither does it mean you must be what? Tall and dark. No. So you can use this Punnett square to demonstrate this as well. I think we are going to do it. Should we do the classwork here? Or should we just move on?
Let's do
I
the
need your
classwork,
feedback.
sir.
Let's do it.
Let's do the
Okay.
classwork, sir.
All right. Wow. Okay, so we have the Punnett square here for the hybrid inheritance. Look at your far right so you see the keys. Oh, hope this is clean enough, clear enough. Circle means what? Round yellow. Okay, I think I should write it then. Are you seeing the okay? Let me use red. Capital R means what? Round. Small r means what? Raincoat. Okay? Capital R means what? Round. Small r means what? Raincoat. Okay. What again? Capital Y. Capital Y should mean yellow. Why small y should mean what? Green. Have you gotten the information? So you're going to use this now to answer these questions I'm going to ask next. So capital R means round. Capital Y means what? Yellow. So we have parents. Now we have two individuals. Now looking at this area, we have capital R. Okay, let's establish which one is dominant and which one is recessive. So when it comes to shape, that's round and wrinkled. Round is dominant, okay? Capital R is dominant. When it comes to color, yellow is dominant. Okay? Now, this one normally takes being attentive, being careful. Now, these parents, look at the genotype. Capital R, small r, capital Y, small y. Please, this parent is what? How will I put this question
They
now?
are heterozygote.
Yes,
Right?
person is heterozygous, correct. Now, I want you guys to tell me, is it round and yellow or round and green or wrinkled and yellow or wrinkled and green?
It's round and yellow.
Eh? Any other person? Any
Okay.
controversial statement?
Let me see.
What's your question, sir?
Okay, my question is that looking at this individual, the person with arrow on the head, the genotype has been given below, and I told us that capital R means round and round is dominant. Small r means wrinkled and wrinkled is recessive. Capital Y means green and green is dominant. Small y means capital Y means yellow and yellow is dominant. Small y means green and green is recessive. So looking at this individual now, what is the what? Phenotype.
The phenotype will be round and yellow.
Round and yellow. Alright, that's very correct as well. Now that's the same thing with the other individual too, right? Okay, so law of independent assortment was telling us that each of these genes will be paired, can be inherited independently of each other. So it means we can have capital R, capital Y, or capital R, small y, or small r, capital Y, or small r, small y. So what we did here is that we wrote out every single combination we can have from this gene, okay? And then did the same for this parent here as well. After that, we'll now start pairing them together. We'll now start pairing them, okay? So capital Y R, capital R, you write the two down here. Capital Y, let me point in it. For this one, capital R, you write it down. Capital Y, you still put it there. Come to the next one. There is two capital R there, you write it. Then there is one capital Y and small y, you write it so as well. Now you continue like that, capital R, small r, pen it down. capital Y, put it. Now, after all this, you do the same till you reach the very end. The hybrid inheritance is very crafty, so all of you have to look at it very well. Now, how many of these individuals are round and yellow? How many of them are round and yellow? I want us to know. Just count. Or should we do it together?
Now let's try, then you make your correction.
Okay, that's better. All right. 48. How many of these individuals are round and yellow?
Nine, sir. Mine is 9 around and yellow.
Okay. All right. Any other one?
Sorry, 10. I counted again, it's 10.
Okay. All right. So any other person? Okay. It's like every other person is either they are too shy to talk, or they are still counting, or they are already asleep. So which of the assumptions should I make? All right. So let's just count it together. Mhm. So counting it together now, we have first of all round and yellow. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Now we have 10 individuals that are what? Both round and yellow. So now, how many are wrinkled and still yellow? Let's see if you are going to find wrinkled and yellow. Wrinkled and yellow. Okay, yes, this one is maybe another color. This one is wrinkled and yellow. Any other one I'm missing? So we have 1, 2, 3. 3 of them are
It's
wrinkled
3. No
and
one is
yellow.
missing, sir.
All
Yes,
right,
3.
3 wrinkled and yellow. So which other one now? Let's look at round
This
and
is
green.
round and
Round
green.
and green. Okay, this is round and
No,
Round
green.
and what, sir?
1. Round and
Round
green
Round
and
is
and
what?
green.
1.
And
Eh?
green.
1.
Green.
1, 2.
Only 2, right?
Yes,
Round
sir,
and
just
green.
2.
Remember, round is
3.
capital R. 3.
Round and
Round
green
and
is
green.
2.
Let's count them.
It's just one, I think.
Mm, Now, this guy is round and green.
And
Capital
green,
R,
both
right?
one.
Yes,
Yes, sir.
double Y, that's small. Then this one as well is round and
It's
green.
2, sir.
So it's just 2. So how many is both wrinkled and green?
Just 1.
Just 1. So now we have 10 is to what? 10 is to
10
3
ratio 2, 3 ratio 2
ratio
ratio
2
1.
ratio 1. All right. So, wow. This took time, but hope it was
Sir,
worth
which
it.
one is 2? Which one is 2?
The one that is 2 is what? Round and green is 2.
1 uncle.
The one that is 1 is wrinkled and green. Okay,
Okay.
so I think this is the last light for today. Though. I've explained it though.
Hello?
Homozygous means what? I'm hearing you.
This one you said second filial generation. Is it because the it's a dihybrid cross?
Yes. I'm trying to get your question though.
Okay. Just I saw F2 generation.
Yeah.
Just the difference between F1 and F2.
So what was the question? Sorry.
The difference between first and second filial generation. I cannot differentiate
Generation
them. I
means
don't know if it's true.
generation means offspring. Now let's look at this. This is the first filial generation. Now I think this is not okay. Now, generation means offspring. From your first filial generation, that's your first generation after you meet the two parents. All right. Now, when you mate those offsprings again, you now get your second filial generation.
When I crossbreed the first offspring.
Yes. Now, let me show you the first filial generation, how it's going to look like.
Okay.
Now, so for this
So you said
guys
first
now,
filial generation is what?
let me come. I'm coming. Just a minute. Now, are you looking at this genotype I just wrote out now? This is capital R, capital Y, capital Y. Now, these are what homozygous for being round, homozygous for being red. Now, if you should cross these homozygous parents, two homozygous parents, you are going to get what heterozygous offsprings. Now, what you are going to get is something like this. Now, this is now your first filial generation. Now, when you bring two individuals from the first filial generation, this one and this one, and breed them, you will now get your second filial generation. Is it clear now?
Yes, sir, it's clear. But if we want to put it down, how can we represent it? First filial generation is what? Because, like, from what you explain, if I get it right, you mean second filial generation is an hybrid of the first filial generation. That is breeding two first filial generation together. That is how we get our second filial generation.
Exactly.
Okay. Then how can we now explain first filial generation? Thank you, sir.
All right. I'll answer your question at the end of this class. I'll answer your other questions at the end of this class. Okay. So let me just wrap up with these ones now. All right. I'm
Okay,
taking
sir.
note of the question.
Okay, sir. Thank you. Okay, sir.
Okay. So homozygous versus heterozygous organisms. Homozygous are the ones with what same allele. While heterozygous organisms is what different allele. Now, like I told us, alleles what affect the phenotype depending on what dominance. Now, so I've actually been explaining most of this. Dominant versus recessive traits. Dominant traits are those that are expressed even with one copy of allele. Recessive must be two before they can be expressed. So genotype versus phenotype. Genotype is genetic makeup, capital T, capital T, small t. While the phenotype is the what physical expression of the what genetic what makeup. Now the genotype determines the what phenotype. So this practical is what you have to now carry out yourself. So when you get an onion root tip, now you should observe them under the what microscope, which you don't have. You don't have access to. But now on the practical level, you should the onions, the onion, the root tip is what very active and actively was dividing. Now when you observe it under a microscope, you should what review the chromosomes. Now you can be able to what picture what happens during each stage. You can see your interphase, prophase, metaphase, anaphase, and telophase. Then you can count the cells after each stage of division. So I believe probably your school should have a lab if it's possible to meet your lab director and you can ask if these experiments can be carried out.
Excuse me, sir.
I'm listening.
Please, can you come again with that procedure? Because though I can meet the lab technician in my school and explain, he will give me room to do it. So can you just explain it while I'll do a recording of it, the procedure, so that I can see how I can run it. Thank you.
All right, so that's it. So the the tip of an onion is actively divided. Okay, so if you can get an onion tip and then view it under a microscope, then okay, first of all, when you get it, there's something called tissue processing. That's all right. Let's leave that part aside. Now, when you get the root of an onion tip, ask the person who he or she should help you stain it carefully. Can use H&E staining so they can what can color the what nucleus. Under the microscope, it should be able to what reveal the different stages of cellular what division. Then with everything I've taught you guys this night, from your interface to your prophase, characteristics of the prophase, then you should be able to see condensed chromosome and all. Metaphase, characteristics of metaphase, the chromosome aligned at the equator, then the anaphase division, sorry, migration have started. Then you can take note, this is the anaphase. And for the telophase, you now notice that is what it has reached the end of the pulse. Now, when you observe all these things, you now match them with what you have written down in your notes. So you can be able to what, make sense of how mitosis progress. Now, at the very end, you can now count each cell in the stage. If at the end of division you have just two more new cells, you will know that is what probably one cell gave rise to two. You know that is what mitosis. That's just the process, okay?
Okay, sir. Thank you very much.
Please, the class has ended, okay? So if anybody wants to leave now, you can leave now. If you have questions, you can also ask, okay? I wanted to end the class for those that may want to leave. There's something like this. Is anyone there?
Yes, sir. This is the right the screen. This is the right screen for
Okay,
what he
so
was asking of.
this was a cross between capital R small r, capital Y small y. The phenotypic ratio was 9:3:3:1. So what we got was 10:3:2:1, right? So let's recount our own now. That being round and green, sorry, round and yellow. 1, 2, 3, 4, 5, 6, 7, 8, 9. All right. Count it again. There's a mistake we made there. I think I've gotten a question now. Round and yellow is 9, not 10. Let's count together. 1, 2, 3, 4, 5. I think we counted this, but this is wrong. This one is round and green. Okay. So 1, 2, 3, 4, 5,
6, 7, 8, 9. Then for round and green, we now have 1, 2, 3. Okay, round and green is 3. And then wrinkled and yellow. Wrinkled and yellow. We have 1, 2, 3. And then for wrinkled and green,
That's one.
we now have 1. So now let's check it again. 9 is to 3 is to 3 is to 1. Right? So it's correct. Thanks for pointing out. Okay, so for F1 and F2 generation, please. F1 generation simply means the offspring of the parents, the original homozygous parents, okay? So when you cross two parents together, their offspring is called F1 generation. Now, their offspring, so when you now cross their offsprings, you will now get your F2 generation. Is that clear now?
It's clear, sir. Thank you.